Yes, dear students, today we'll be discussing about methods of gene transfer, the second part of this lecture. Yesterday we had a fruitful discussion regarding the gene transfers methods using the viral vectors. Today we'll be using the discussing about the different physical methods in this regard. So directly moving towards the slide of which we have to discuss. Let's hear. Yeah, this is the main slide. So we have non-viral vector methods. One is physical, another is the chemical. We have in physical, we have electroporation, gene gun, and while in chemical methods, we have polyplexes, lipoplexes, nanoparticles, polymeric mass, silase, and dendrimers. So directly coming to the electroporation. Uh, Electroporation is from the word you can easily judge it, that electroporation means you just give an electric impulse, and poration means to create pores in the cell surface. So basically it's a momentary exposure of cells suspended in DNA solution to a high electric field. Normally what we do is we have a cuvette, small, like, have you seen, all of you have seen the cuvette of our normal DNA measuring use spectrophotometer. Similarly, there is a type of cuvette in which there are two aluminum electrodes, and the space within the two electrodes is in few millimeters. We put the cells along with the DNA to be transferred in that solution, and we give us a high voltage current in small impulses, 2 impulse, 3 impulse. And what happens with this impulse is that it created pores in the cell membrane, and it when the pores are created, actually these assist the DNA to enter into the cell. So normally the biggest advantage is it has a high transfection efficiency, means the efficiency of transfection is much more, and we could handle a various type of cells, as well as cells from the different organisms, and the various types of cells could be handled using this electroporation, from microorganisms to plants and humans and animals. What type of cells we have from all these organisms, we can handle those, and we could have a transfection efficiency from all these things. And secondly, what is that is that disadvantage with this electroporation is that due to this higher voltage, there is a significant number of cell deaths, or damage is so high to the cells that most of the cells die. So we have high transfection efficiency, but at the same time, the biggest disadvantage is the damage to the cells. If we have a look on how the electroporation takes place. So if these are the cells, and we have this DNA outside, and we give a current, so pores are like created in this manner, and the DNA or the plasmid which you have transgenic recombinant plasmid you have made would enter through these pores into the cell cytoplasm, and then to the nucleus for integration. And later on, when the pulsing is stopped, or electric current, after resting condition, the cell membrane would return to the original state. So electroporation just creates pores. Rest of the things is done in an autonomous manner, that DNA enters into the cell. Other is physical method is the gene gun. You all have heard this name, gene gun. Normally we use a helium gas under pressure to transfer the material. We also call it as a biolistic gun. The method is that we have coated gold particles with DNA. That, in other words, gold particles are coated with the DNA, and we put them on the load macro carrier, which is placed in the gun plunger. And when we release the pressure, this would break, and this would bombard under high pressure on the cells. And when the cells are bombarded, you could have see that under pressure, these gold particles along with the DNA enters in the cell, and transfection or transformations takes place depending upon the type of cells you are bombarding. If you are bombarding the microorganisms, it would be transfection, and if you are again bombarding the plant cells or eukaryotic cells, is transaction. So moving further, gene gun is also called as biolistic transformation. Advantages is can be used in any cell type similar to electroporation, and the disadvantage is damage of a significant number of cells, low transfection efficiency as compared to the electroporation, and reproducibility problem that you might have. Sometimes you could have uh, a number of the number of transfection efficiency is high, and while in other terms you could have less reproducibility. So
सेम सेम पेंसिल इकट्ठी रखो.
in gene gun, the reproducibility problem is there. So we have to be very sure that what we are doing. And moving further from gene gun to the other part, that is, one is another method is calcium phosphate method, or for gene transfer. It is also called as co-precipitation method. We all know our DNA is a macromolecule and negatively charged complex, and what we do is we make actually complexes of this DNA, negatively charged DNA, with a positively charged calcium phosphate crystals, and these complexes are then taken up by endocytosis by the cells. The advantages of this is it's an inexpensive and simple to perform with high transfection efficiency. The biggest advantage is cytotoxicity, that it could kill few of the cells. But coming to the point, what we do is we have two buffers. We have a heap buffer with sodium phosphate containing heap buffer, and one we have a calcium chloride along with the DNA. When we mix these two solutions, then calcium phosphate crystals along with DNA complexes are made, and we put those solutions back into the medium where the cells are present. And with the passage of time, what happens that those cells uptake those crystals of calcium phosphate along with DNA by the endocytosis phenomena, and gene transformations takes place, or gene transfers takes place. We have already discussed the endocytosis phenomena, how these endocytosis takes place. So it's just a elaboration for you all. Moving towards the other part of this, that what other methods could be used for gene transfer. This is the upcoming and very field of biotechnology, which is actually nanobiotechnology. If we just want to define few of these terminologies, so nanotechnology is actually the field which actually manipulates with the matter between the size range of nanometer size, that is 1 to 100 nanometer. While if you want to define what's nanomedicine, is actually its application of nanotechnology in diagnosis and therapy. So such type of applications are called nanomedicine applications of nanotechnology. Coming to the nanopharmaceuticals, so these are actually therapeutics consisting of at least two components, the drug and the carrier nanoparticle on which the drug is placed in the size range of 1 to 1,000 nanometers. So we have biopharmaceuticals, which are actually, we are developing, scientists are developing various types of nanoparticles, which are actually acting as carrier for drug molecules. And later on, the nanobiotechnology is the use of nano materials in biotechnological products. So moving further, This diagram lists the nanoscale, relative size, and detection devices that human eye could see up to 1 millimeter size, or light microscope could see a cell or a hair cell. Electron microscope could see up to 1 micrometer, and after that we need electron microscope for small nanometer size particles, which includes bacteria, virus, or DNA, or small molecules, atoms. So this is the explanation of nanoscale. And in the biological fields, or chemical sciences, normally plant cell is of almost 10 to 50, 60 centimeter, animal cell micrometer size, while the DNA is almost 100 nanometer, and one helix turn is almost 10 nanometer. While we have a almost zero point 0.3 uh, nanometer difference between the two bases. We have already discussed it. So it varies from, so size is the basically the hub of this nanotechnology. Now coming to the nanoparticles, and few of these are these 3-4 are actually used in transferring the genetic material to the cells. Number one is the polyplexes, then lipoplexes, then polymeric nanoparticles, and polymeric micelles. We will go through all these one by one, and let's have an overview of this. What's polyplexes? Polyplexes are actually complexes of cationic polymers with the nucleic acid. Now coming to the cationic polymers are all positively charged, and nucleic acid, we all know it is negatively charged. So what happens, we make, due to these positive and negative interactions of these polymers and nucleic acid, we form complexes, and these actually complexes, which are named as polyplexes, could be used to transfer the genetic material from into the cell. Polyplexes formulation is normally regulated by electrostatic interaction, which is normally in fact affected by the pH of the media, ionic strength, and cationic density. What is positive charge density, charge density on these cationic polymers? That actually determines the polyplex formation. So these are the two major examples of polyplexing forming polymers, that is polyethylene amine and the polyallycid. Sorry. Polyallycine. And what happens in polyplexes is we have a DNA which is negatively charged, you can see here, and we have a polyethylamine polyplex which is positively charged along with few ligands. And what happens, the DNA is complex with it, and to create a neutrally charged polyplex with a ligand, and this ligand would attach to a specific receptor on the cell membrane, and then this polyplex would be internalized, and the DNA would be transferred to the cell. Advantages of polyplexes is easy to formulate, and they are simple chemical modifications, while on the other hand the disadvantages is cytotoxicity and stability problem, that the life of these polyplexes is not long. You have to be sure about the stability, about the medium ionic strength, so that they remain for a longer time and you can use them. Moving towards lipoplexes, these are actually complexes of cationic lipids or liposomes with nucleic acids. In other words, what we normally say is these are actually negatively charged lipids which mix with the nucleic acid, which are, uh, these are positively charged lipids which mix with the negatively charged nucleic acid to make complexes. So lipoplex formation is regulated by similarly electrostatic interaction, and lipid constitutes the carrier status in this lipoplex. Normally, transfection using lipoplexes are called as lipofaction, and we have different types of commercially available lipoplexes, which is lipofectamine or lipofact. This is the process that we have a lipofaction agent, and we have the plasmid vector DNA. We create a complex and then put it in the medium where the cells are growing. And with the endocytosis phenomenon, these liposomes carrying the DNA, they would be integrated into the, or they would be endosomed into the cell. Examples of lipoplexy is lipofectin and lipofectamine. These are the two main commonly used lipoplexes, and they are commonly used for doing gene transfer. Coming to their advantages and disadvantages, advantages is the versatility of these lipoplexes, protection of DNA and with the cell membrane, and biocompatibility, that they could handle different types of DNA ranges. And disadvantages, they are labor intensive, expensive, issue of stability, clearance by reticuloendothelial system. As compared to the other plexes, they are more easily cleared by the reticuloendothelial system. So this is a bit, but I have been using this lipoplexes during my own work. Certainly, these lipoplexes and polyplexes have a transient expression, because in most of the times these don't get it. These the DNA which we have transferred is don't get into integrated into the nuclear genome, but we have good results. So it's not a big deal that lipoplexes don't work. Now coming to the polymeric nanoparticles. So nanoparticles are made of polymers, and natural polymers which are normally used are chitosan, albumin, and gelatin, while coming to the synthetic polymers which are used are polylactic acid, polyglycolic acid, or combination of both these polylactic and polyglycolic acid, named as polylacticoglycolic acid, which is PLGA. So these three polymers are used, or above natural polymers are used to make polymeric nanoparticles. Procedures about these, how to make these nanoparticles and their uses. I have downloaded almost 2-3 papers for you, and will share with you all this stuff so that you could have a more detailed overview of these nanoparticles, how they are made. Presently, interest of my lecture is not that. I'm just making you aware of what are the different things which could be used for gene transfer. We are not going into the details how these are made and how we use them. So polymeric nanoparticles have few advantages, that is biocompatibility, biodegradability, loading of more than one gene or drug, while in terms of disadvantages, the tedious preparation, degradation of DNA during preparation, and it can't be easily modified. So these are the few advantages and disadvantages of these polymeric nanoparticles. Moving further, the last but not the least is the polymeric myceli. This is actually, from the structure is, you can easily see there's a core, and then there is a outside corona or shell of this myceles. These are actually made of amphiphilic polymer chain that self-assembles into nano-sized spherical structures in aqueous medium. Amphiphilic polymers are such polymers which could have both hydrophobic and hydrophilic properties. A few parts of those polymers are hydrophobic, and few parts of these are hydrophilic. So these polymorphic myceles are also produced, and advantages: biocompatibility, biodegradability, easy to prepare, chemical modification easy. But the biggest disadvantages with them is their unstable nature, and they have to be made fresh all the time to be used for gene transfer. So this is all for today, and these are the learning output of this lecture, that in last 2 days what we have identified is, or discussed is, two main methods of gene transfer, that is the viral and non-viral methods. We compare the different viral gene transfer vectors and what is the best for any therapeutic strategy we need. Then we differentiated between the physical and chemical methods of gene transfer, compared the different narrow carriers, and what are the different non-viral methods which could be used as a therapeutic strategy for gene therapy or gene transfer. So this is from my side, and in terms of methods of gene therapy, gene transfer. And we will be having further lecture on Monday in this regard from the other course outline. Thanks for today, and we will be having a discussion further.